series of environmental studies research presentations. If you are here and wanting to get extra credit for your class or are part of a class where you need to let me know that you're here, here's the sign-in sheet. Put your name on it and your class. That would be wonderful. So for the new people here, I'm Dr. Lynn Trulio, and I'm chair of the Department of Environmental Studies, and I want to welcome you. And we are very pleased today to have an expert on plants of California, Dr. Tom Parker, and he is a professor of biology at San Francisco State. I've been there since about 1980, I guess.
What's that?
1980?
Yes.
Yeah, I know. That's fantastic. Students have benefited from me being there, and every once in a while we'll have a Tom Parker sighting here, and so we're thrilled to have him here today. He's going to tell us about manzanitas, and which is a fantastic, if you've seen them hiking at all, around here through the shrublands, it's an amazing plant and a very diverse taxa. And so Tom is an expert in the ecology and evolution of these plants, and he's going to be telling you about these aspects of this genus of plants and basically how they became to be so diverse in our region. So he teaches classes on plant ecology and community ecology, fire ecology, and also graduate seminars at San Francisco State, and has written a number of papers on Arctostaphylos as well as on wetlands. And so I want to have us all give Tom a nice welcome. And actually, before Tom starts, I just wanted to welcome Rod Myatt from our biology department, who's a long-time friend of our department. Take it away.
Thank you for the invitation. It's always great to be invited someplace and to see old friends again. It's also really easy for me to want to talk about Arctostaphylos. This is a group I don't merely enjoy doing research on, but I actually love doing research on them. So it's more of a love affair, and just for those of you who want to do anything in your life, just hope that you fall in love with whatever it is that you're doing. So that's what this group is. It's a really beautiful, highly diverse group. It's the most diverse woody plant genus in California. Probably the whole West, but at least in California. I tried to think it was the most woody, most diverse one in all of North America, but man, you get into oaks and you include the rest of the U.S. and all of a sudden it gets messy, and then there's rubus. But at least in California, this is the most diverse group. They are absolutely lovely, and if you're out there at the right time, they are so heavy with perfume from the nectaries that you think you're walking around in a honey jar, right? And of course, the red bark is the classic way to identify the genus, even though 5 or 6 of them lack that character, just to confuse you. Here's where the species richness is dominated. So the California Floristic Province is basically it. There's 5 to 7 species really outside the province. Not very many. You're only looking at 2 to 3 species here, 2 species there. Arizona has 3 or 4, and this is just one species, Arctostaphylos pungens. So most of the diversity is here. Of course, there is one species that's worldwide in the circumboreal area, Arctostaphylos uva-ursi. So it's a great genus. There are 27 people who have named taxa in it, and we've got Linnaeus and De Candolle and all sorts of famous people, including von Humboldt, because of their explorations and distribution patterns. A lot of the reason for their diversity is they've been able to adapt to basically California, and that means a lot of topographic variation with a huge diversity of soils and a huge diversity of climates as a consequence. So this is a granite outcropping where Gabelonensis is found near Fremont Peak, and it's only found on this granite outcropping and one near the Pinnacles. very large populations, but those are the two granite outcroppings and the manzanitas, and that's the only place you find that plant. Montairensis is a shrub to small tree on Monterey Mountain in northern Santa Cruz Mountains. It's a beautiful plant as well, because I can hike to it every day. It's in my city. Montana is found on serpentines, restricted to them. So a lot of different kinds of soils. In the middle of the Central Valley, there are the ion oxazoles. Do you guys know about those? Old tropical soils that have been uplifted, extremely acidic and nutrient poor. And this darker plant is Arctostaphylos myrtifolia, which is only found basically on those soils. There are two southern populations on a slightly different kind of soil. And the lighter color is Arctostaphylos viscida, the white leaf manzanita that's pretty widespread. Not very far from here, if you drove over to Bonnie Doon and got on Martin Road, you'd find sandstone outcrops that degrade rapidly to sand. And on these sand dunes are Arctostaphylos sylvicula. That's this lighter colored one. And that's really an endemic to those sandy outcrops in that part of the Santa Cruz Range. Good. I turned my phone off. I also wanted to point out that there's a huge diversity in terms of the climates that they're in. There is a maritime set of species that's in the coastal fog zone. And those species are pretty much restricted to the fog zone, and they include a lot of the very interesting taxa with auriculate leaves that are very odd. We're actually doing experiments to see if the auriculate leaf morphology increases the rate of stem flow from fog drip. So it's kind of a fun group to play with. Others that are also restricted to the fog zone include sensitiva and numularia that have more normal looking leaves. You go to the interior areas. This is in the interior side of the Santa Lucias. These are dry and hot locations. You can get into the Sierra foothills where it's really quite dry and warm in the summer. Another endemic to that area, Nissanena, found only in a small part of the central Sierra foothills. Viscida, again, nice widespread one. And the South Fork of the American River, if you know the area. We're hoping that burned. Nevadensis is another widespread species, but this one's high elevation. You only find it above the snow line. So you have to go look at it in the summer, and that's that prostrate green mat that you see. Its common name is pine manzanita. And of course the green leaf manzanita, also around the same elevations. This one is widespread in California. It is a resprouting species in California, but it's also widespread in the Rockies, and it's not a resprouter in the Rocky Mountain areas. So it's a little bit different. So in addition to soils and climate, wildfire is probably the most important piece to the evolution of this group. It's commonly a member of chaparral or edges of places that have high frequency fires. And the trick for this group is the fires have to be high intensity. So if you ever hear people say that chaparral is senescing, it never senesces, but it always has high intensity fires. You can't have a low intensity fire when all the fuel is right at the ground level. Right? So you always get that killing fire. And that's a way that they are able to maintain themselves with the mixed evergreen forests on the coast here or the conifer forests in the Sierra. As those trees begin to invade chaparral, you get a fire that knocks back the trees, so they get to maintain themselves. The forests have actually increased in the Sierra Nevada, according to geographers who have studied aerial photographs, that fire suppression has permitted conifers to constantly invade in the chaparral areas. There's a mutualism that is involved in that. Arctostaphylos has Arbutoideae mycorrhizae. You guys know what mycorrhizae are? A fungal mutualism in the root zone. And they share about 80% of their taxa, which is hugely diverse, with conifers and the Pinaceae. So it's really easy for a pine or members of that group to invade Arctostaphylos chaparral because they can link into this hyphal network that's underground. And that permits them to survive dry summers, even though they shouldn't, because they can obtain water and carbon through tapping into the mycorrhizal hyphae. So they're basically being supported by Arctostaphylos. What happens after those fires, though, is it looks pretty devastating. People don't like that. Ecologists love it, because we know that the dynamics are now going to start. And the dynamics include the germination from dormant and persistent seed banks of all the members of Arctostaphylos. So there are 3 genera in California that have this method of surviving fire. Arctostaphylos is one of them, with really large seed banks that are buried in the soil. Ceanothus is another one, but its seed banks are quite small, but more than enough to maintain the populations. And Chemise, or Adenostoma fasciculatum, is the third one. About a third of the taxa have a wooden structure that's expanded at the base of the plant, and they can resprout from that. And if you're reading keys, those are called burls. It's very dense wood that's just filled with dormant buds. And they basically survive wildfires. You can see you're going to end up with a ring of plants through time, and as there are multiple fires, the ring of plants turns into a literal ring of plants. But if too much carbon and litter build up in that area before they spread out a lot, then a really high intensity fire can just ash right down into the root zone and undercut them and burn them out from the inside. So as you're walking through post-fire stands, you have to kind of watch out, because there can be a lot of holes in the ground that were woody plants that ashed all the way down. A year later, you end up with large plants from those resprouting species. A lot of those plants, though, don't resprout, and they're just still pretty small plants after that first year. So here's a first day after a fire. You can see the burl. These guys are termed facultative seeders because they do have that persistent soil seed bank, and they do recover new individuals from the soil. So they can recruit new individuals post-fire, even if they do tend to resprout and survive. This is a species, Arctostaphylos canescens, that lacks burls, and so they're completely killed by fire. And they're referred to as obligate seeders because the only way their populations recover is from germination of the seeds in the following spring. So there's actually a lot of problems in the Santa Monica Mountains right now because they've had fires, but they've had almost no rainfall for 4 years. But last spring they did have 1 rain, and it came in March. But it was a single rain. And a lot of seedlings came up because the plants are dying and have allowed the soil to be heated. And as a consequence, a lot of, at least Ceanothus seedlings have come up. The manzanitas are just dying. They have more complex germination. So it can be difficult. I want you to have this question in the back of your mind for the rest of the talk. Where did the obligate seeders come from? Think about what a life history method that is. All the adults die in a wildfire, and all the dice are rolled with seeds. So these things have to germinate, they have to survive herbivory, they have to survive a lengthy summer drought. Ours is the worst Mediterranean climate in terms of droughts. We have the longest, most intense summer drought. How did they ever come up with this as a viable strategy? If you look at other members of their close relatives, they all resprout, so this is the new evolutionary innovation. And it's probably why there's so many of them, because if you also think about it, they roll the dice, all the seedlings are coming up, they're all genetically distinct, and whatever the climate or other environmental conditions are that year, it's going to push the genetics of that germinating population. And so you'll have an entirely new group of individuals with each fire. For those resprouters, most of them survive, and so the genetics of the population remain very stable from one fire to another. Only a few individuals shift. So think about that in terms of rapidly changing climates. The obligate seeders can actually drift around with climates, and the obligate, or the facultative seeders, the resprouting species, really take a long time. I've actually modeled it. You need to have at least 30 or 40% death before you get more rapid change. And that still takes thousands of years. Okay, the other piece I want you to think about is dispersal. These plants are in a habitat that's ideal for them, but they do disperse. So where do you disperse? Well, the trick is you have to disperse not horizontally, although coyotes and bears will help you do that for climate change. But the most important piece is you have to disperse vertically deep into the soil. Those high-intensity fires produce killing temperatures for the first several centimeters of the soil. So if your seeds are in the upper part of the soil, the first 2 inches or so, they're not going to survive the heat pulse that comes from the fire. So they have to get down pretty deep. For ceanothus and chamise, those have very small seeds, and it's not too difficult for their seeds to work their way down through the soil. But manzanitas have pretty large fruit. So it's a very different kinds of process. So we're going to focus now on a mutualism that's very critical for manzanitas in the context of wildfire. But you also have to start thinking about how long this mutualism must have been going on. So we have those 2 different life histories. All the species produce dormant seed. Those seed won't germinate except with chemicals from smoke and other physiological requirements we haven't quite figured out. You can go to the grocery store, buy liquid smoke. You guys know you can do that? Dilute it very heavily and you can get a little bit of germination, but not much. So we know there's other pieces to the puzzle. Some of the species are killed by fire, and those are obligate seeders, and some resprout. So these are about 2/3 and these are about 1/3. The key piece here is that those seeds remain dormant from the time they leave the plant until there's a wildfire. I showed this picture because I thought you'd be interested that they already have red bark right as they germinate. I think that's cool. Right? It's these kinds of things that infect me with love for the group, right? Okay. Let's think about the structure of what these soil seed banks have to look like. So if this is the soil surface, and this is depth in the soil, and this is density of seeds, that's what a typical seed bank is going to look like. Most of the seeds are going to be shallow, and they're going to be killed by fire. Only a few will be deeper in the soil column. So if you have a high-intensity fire, you're not going to have much survival. And if you have a low-intensity fire in that location, it'll have a higher density of seeds surviving the fire, right? So there's a lot of heterogeneity in any one fire because of topography and other issues. So how do the fruit and the seed get deep enough to survive these fires? They're in a variety of soil types with different types of textures, but nonetheless the fruit, the smallest fruit are 2 to 3 millimeters in size. It's pretty small, but it's much larger than particles of soil. And the larger ones are up to 15 millimeters in diameter, and they're globular, they're globose. How do those things ever get down into the soil? You can see how big these things are. They're dry, pulpy drupes. Coyotes, bears like them because they get a lot of vitamin C from it, but the seeds pass through them unaided. And the seeds are just way too large. If you pull all those fruit apart, these are typical sizes, but that's still pretty large. And some of them have all the seeds fused together into a single structure, and those are way too large. There are animals that live in the chaparral, and these are just the granivores. I like the granivores. I also like the predators. I had a student working in a chaparral stand. He was from Germany. And I said, you know, I don't like you going out there by yourself. And he's 6'4", a really large, strong guy. And it's like, no, there are mountain lions where you work. He's like, oh, you're kidding me. I said, no, take one of the trail cameras and put it on the road. So he did eventually do that. He got skunks and bobcats. In fact, the coolest one was a bobcat that laid down in front of the camera perfectly and then started acting like a kitty cat. He kissed himself and rubbed his head. And I was like, I didn't know they did exactly like kitty cats. But we never saw the mountain lion, so I had to get him onto a website called santacruzpumas.org or something like that. So he could see that they go right through his research site. These guys are probably not important for Arctostaphylos because the seeds are too large and they start off as fruit, but they're really devastating for ceanothus. On the other hand, these guys can be pretty deadly. They're major seed predators. They're desperate to compete with one another, so they harvest as much as they can. They run off and they bury it. And they run back and harvest more and run off and bury it. They're called scatter hoarders. So they'll bury one to multiple fruit in a hole at a time. And the trick is, how deep do they bury it? Okay. So the granivores are either consuming it immediately, so the birds would, for example, immediately consume Ceanothus. The rodents might eat, or they might cache some of the seeds or fruit of Arctostaphylos. So this is sort of a conditional mutualism, because it's not a mutualism if they eat all the seed they disperse. Okay. It's just seed predation. So they have to miss some of them, and that's why I like rattlesnakes and coyotes and bobcats, because they like to eat these rodents. So you have this kind of weird love affair with rodents after a while. You love them for doing important work for manzanitas, but you want them to die at the same time. So it's a very strange research place, right? It's also considered a diffuse mutualism because it's not this tight little thing. It's not like one rodent species per manzanitas. It's every rodent that ever has cached fruit. Doesn't really matter. Their behaviors are the same. They do this with conifer seeds or any other seed that they could find. And the nice thing is they rely on smell. That's also a complication, because after they bury them, they can find their friends' caches. And they will dig up their friends' caches and go bury them someplace else and hope they don't find them. So they have this little competition amongst themselves. So here's a post-fire stand with the Pacific Ocean. You guys are close enough so that's not impressive to you. I always like to go to places where they don't have that. You find things like this. That's not a mowed down single individual. That's a cluster of 20 to 30 different seedlings all arising from a single rodent cache. Okay. You can see it's pretty big. If you go harvest data from lots of fires, this was from 3 different fires, 5 different species. And this is the percentage of seedlings from the caches, and this is stem diameter remaining, which is basically fire intensity. And what you see is you get a constant pattern, regardless of whether it's from the Sierra Nevada or the coast ranges, fire intensity and the percentage of all the seedlings that are coming from rodent caches. Basically, as fire intensity increases, the seedlings that you're seeing are basically coming from rodent caches. That's because if they did somehow get into the soil without rodents burying them, they're way too shallow. And so as long as it's a low intensity fire, some of them can survive. But as you get to high intensity, you've got to be deeper and deeper in the soil. Somebody's got to bury you. So thank you, rodents. That's important. So we wanted to actually make sure our rodents do this. So we had to do an experiment. And that's actually me. We put a petri dish filled with fruit that we collected on site, and we put a pizza pan down where we had glued sandpaper, and then we covered the sandpaper with fluorescent powder so that the rodents would have to walk over fluorescent powder to get the fruit, and then they would have to walk over it to go and bury it. And then we could follow fluorescent trails and go back and dig up their caches and measure how deep they were. We actually did that. Here are some of these are actually footprints of rodents on these lovely red stems. I'm glad I chose fluorescent green. And here's a tail drag, so it was an easy one to follow as examples. So we actually found around 50 caches, and I was working with a friend of mine who's actually older than me, and we worked, and we found 2 caches, and we're super proud. But of course, the 2 techs found 48 caches. So you want young eyes out there? It really helps. Here's the results. Here's the soil surface. So the depth, the average depth was about 4.5 centimeters, and that's well below a typical kill zone. And if you look at the distribution, there's a lot of them way too shallow. They would be killed, but plenty of them deeper into the soil column. So it's not going to be an issue there. How many fruit did we actually find in these caches? So the average number was 4 fruit. And that's going to vary depending upon the composition of the rodent community that you have. Really small Peromyscus aren't going to stick too many fruit in their cheeks, but there's chipmunks and larger animals as well. We only did this overnight, so it's only going to be the nighttime rodents that would be acting. But for fruit with an average of 6 or 7 viable seed per fruit, gives you 28 or 30 different seeds that would be coming up out of the cache. And here's the range. Most of them are 1 or 2 or 3 fruit. And that's because this site happened to have a lot of small rodents, but there were a lot that were larger numbers. I want you to think about something else. Now we've got our seed in the soil, and a heat pulse comes down, and it has to be able to survive that heat pulse. And the seed now are enclosed in a woody structure, the endocarp of the fruit. So that's just kind of like a nut. You ever open a pecan or a walnut, you're opening up the endocarps, the woody part, and the seeds are on the inside. So that woody structure can protect them to some extent, just like the bark of a tree can protect trees from fires to some extent. But let's think about how these seeds are actually in the soil, because this was a question I was interested in. Here's that same seed, but it's actually in a cache, covered by a whole bunch of other seed. So if we only focus on the bottom seed, does the cache disrupt that heat flow so that you could actually have seed up in that danger zone, the kill zone of the soil, and actually have a little bit more increased survival? So we did that test, and we did it by modifying an oven in the lab. We put in an infrared heating block that can generate up to 1,000 Celsius, but the oven can't hold 1,000, but it was able to hold 600 Celsius, which is super hot. And the cool thing is the door was held by magnets, but the magnets would give up magnetism at about 300 or 400 Celsius, so the student had to sit in a chair with his feet cold. This project has had lots of cool, bizarre stories, right? So what did we find? First of all, we found that there's very low viability in Arctostaphylos. This is no mortality, so you're only looking at about 45 to 50% viability on these seeds. So there's a major statistical issue if you're trying to say things are significantly different. And by the time you're around 125 to 150, there's no survival. So at somewhere around 85, you start losing viability. 85 is not that hot, right? But you start losing viability and you're completely gone by 150. So the killing depths are usually that, and we buried caches 2.5 centimeters, or less actually. And we got a 10% absolute increase. These are 2 sets of caches. And that's about a 25% increase because of the low viability of the seed bank. So caches do increase seed survival just by themselves. So even if the rodents aren't smart enough to bury them deep enough, at least some of the seed will still probably survive. So the manzanitas get it both ways. Thank you for burying us deep enough, and thank you for burying us in a cache. So we may have problems when we germinate, because I have to now fight with all my siblings, but at least I survived the wildfire. So that's the implications of rodent caching, is some of the seeds are buried deep enough, and some of these are differentially surviving very high intensity wildfires, as you saw from the field data. And caching can actually increase survival for some of the seeds within those caches, even though they might be in the kill zone of the heat pulse from the wildfire. That's kind of cool. So I put a tagline in the title to lure in zoologists, so I don't know if that was effective. But I've got these plants manipulating the rodents, but it doesn't look like it. It looks like the rodents are doing all what rodents do. Just collect fruit, bury it, eat all the seed that they can do. But I'm thinking that manzanitas are manipulating these guys. And I'll give you a little background to convince you that I think that's what's occurring. So they're in the subfamily called the Arbutoideae of the Ericaceae. So that's a set of genera that's fairly primitive in the Ericaceae, and you might know that plant family for blueberries and rhododendrons and cranberries and things like that, but it's really a super diverse family. Okay? Arbutoideae is named after one genus, Arbutus, which is the madrone. You probably do know the madrone, or at least the strawberry tree that's probably planted on your campus. That's actually from Europe. So here's Arbutus manzanita on the bottom, and the strawberry tree, Arbutus unito, at the top. This one is widespread on the Pacific coast. Mexico has another 8 or 10 different Arbutus species. Arizona has an endemic. One Mexican one crosses the border into Texas. The Mediterranean area has 3 different species, as well as a large hybrid zone between 2 of them. We also have a genus called Arctostaphylos. And if you remember the last one, those are large succulent fruit. The seeds in the Arbutus, or the madrones, are kind of like seeds in tomatoes. You try to crush them with your teeth and they squirt to the side, so you can never do that because they've got a little slit covering. Okay? And so they just pass through animals. And those are bird dispersed. And I always wondered about that. I always watched, you know, nothing's eating these damn fruit, and then a flock of band-tailed pigeons would come in and scare me to death and start eating all the fruit. If you've ever been in the field when they fly off and you weren't expecting them. Arctostaphylos also has that succulent outside, and it's bumpy. But the seeds in this case are now enclosed in an endocarp, just like manzanita seeds, and they're all fused together. But they're quite small, just like Arbutus seeds are quite small. We only have one Arctostaphylos in California, in the southern part of the state and on some of the islands. But it's pretty diverse in Mexico. So it's another group that is diversified to the south of us. We do have Xylococcus bicolor, which has got the name Mission manzanita, and it's really common from Santa Barbara to San Diego and into Baja California. It's now got a dry fruit, as does Ornithostaphylos, which is also only San Diego and northern Baja. In fact, it may not be in San Diego County anymore, because it was only just across the border in an area where Homeland Security keeps building fences and destroying sites. They both have these dry fruit. The seeds are fused together just like Arctostaphylos, but again, very small. And here you start looking at Arctostaphylos, and you've got things up to 15 millimeters in diameter instead of those 6 or 7 millimeters. And a huge diversity of structures. So here's our Arbutus seed and fruit. Arctostaphylos one, not quite mature. I thought about coloring it with a Marxolite to get the right color, but it didn't work, so a green fruit. And here's some variations of Arctostaphylos fruit with the seeds that came out of them. So what you can see is there is an evolutionary trend in morphology just within the fruit in this particular group. Arbutus has fossils that go back over 50 million years, and Arctostaphylos has fossils that go back about 15, a little bit more. So that's the young baby in the group. And according to the DNA work we've done, it's one of the most recently evolved. But we're going from succulent bird dispersed things to a dry exocarp and mesocarp, the outer part of the fruits, and it's mealy on the inside rather than succulent fruit. The kind of thing that if you ate it, you would go, what the hell am I doing, and start spitting it out, because it's that kind of pulpiness. Huge seed. So here's what an evolutionary tree looks like. Ironically, these guys are more closely related to the Mediterranean madrones than to the North American ones. And we've had that confirmed in a second study, so it's very weird. And we're going to ignore Arctos, because these are all Mediterranean climate species. And if you look at their mode of dispersal, you shift from bird to mammal. By the time you get to Arctostaphylos, you go from all transient seed banks to persistent in Arctostaphylos. And transient just means the seeds disperse and germinate readily, usually within months. They don't last too long. So think of an acorn from an oak tree. That's a transient seed bank. The seeds need to be buried pretty quickly or they lose viability, and then they'll germinate either immediately or within months. Right. These, on the other hand, don't germinate until a wildfire stimulates them. And because of that, they have very different life histories. All the others are obligate sprouters. They disperse fruit in the fall and winter. Their seeds germinate in the middle of winter, that kind of thing. Arctostaphylos matures fruit in the summer. And it disperses fruit at a time when the rodents will bury them prior to the winter rains. And they won't germinate without the stimulus of wildfire. And as a consequence, huge diversity in this group, especially because of the obligate seeding. So there seems to have been reciprocal evolution between Arctostaphylos and rodents, and to some extent in the other genera in the groups. Rodent caching, if you think about it, that had to evolve a long time ago, and it had to be very consistent. It's going to be an evolutionary natural selection force, but to do that, it had to have been there to be able to provide that. It had to be consistent. Once rodent caching was consistent, then seed dormancy could evolve. And it would be selected for if it was favored to establish individuals post-fire when there's no competition from other plant species. You can imagine how rapidly that would be selected for. And then you would end up with these pulse-fire pulses of establishment, and then obligate seeding could evolve. You can't evolve obligate seeding without persistent soil seed banks first. Otherwise, you just lose your population. So there are a sequence of steps that had to have occurred. And it looks like all the changes are happening in Arctostaphylos, right? And in that subfamily, the rodents are just doing what rodents do. So how is it that plants are manipulating rodents? So if you let me tell you that I think the rodents are just operating based on a behavior model called optimal foraging. And in that model, they only forage as long as the total handling costs permit them to gain more energy than they expend. Otherwise, they're using more energy than they're getting, and that's not a viable behavior. So they would quit foraging to, they also quit foraging if there's any hint of predators in the area. Right? So you have to kind of incorporate, are they getting enough energy and are there any predators nearby? We've had trail cameras up with time stamps on them, and if a bobcat walks in front of our cameras, we don't see rodents again for a day or two. They don't come back. They can tell that there's been a predator there. Now, manzanitas provide a lot of seed, and they do have pretty large seed banks, but they're dumping tons of seed almost every year. And if you've got wildfires that only occur once every 50 to 130 or 40 years, that's a ton of seed that's being dumped out there to maintain the rodent populations. So half of those seeds, though, are empty. We just saw in the experiment on heating. Now, these empty seeds increase the cost of foraging for rodents because if they pick up an empty one and don't realize it's empty and chew it open, they've burnt a lot of energy digging it up and breaking it open and don't get anything for it. So that should increase the seed density in the soil more than you might expect if these were wonderful things to eat. I have a feeling that they're only sort of okay to eat, but because there's nothing else to eat in the area, they still eat them reluctantly, but happily. Plus, there's another little trick. A lot of the seeds are fused together. If you think back to one of the earlier pictures, a species that's really close to your campus, the bigberry manzanita, Arctostaphylos glauca, all the seeds are fused completely into a single structure. Right? So all 7 or 8 are completely fused, and that would be over here at the far side. Some of them are always independent. So Arctostaphylos viscida and Arctostaphylos canescens always produce fruit where all the seeds are independent of each other when they're dispersed. But most species actually have one independent, a couple fused together, all in the same fruit. And that varies a lot. But what's kind of cool about it is the proportion of viable seed increase as the proportion of fused seeds increase, which shouldn't be terribly surprising. If almost all of your seeds are in a single structure, you better have something viable in there. But this is the important piece. These are completely viable, every structure, and these are completely empty, every seed. So some of those completely fused ones have nothing viable in them. okay, or a 1 or 2. So they have to work their way through to find that one viable one. And to me, it's this scatter shot that's unpredictable for the rodents, that increases their handling time, that's going to get them to say, I gotta move on. I'm just failing to do that. Here's the non-sprouters. They have a higher viability, but it's the same kind of pattern. Except nobody's completely at 0. So here's the projection or prediction that we're running right now to test this idea. If the seed or propagule is really large, it should be easier for the rodents to find them. And if they're all fused together, which is when they're really large, then there should be something viable in there, so the rodents will probably reduce the seed bank density pretty effectively. So it'll have a low density. It'll still be pretty big compared to Ceanothus. If the seeds are kind of small, on the other hand, there should be a pretty high density, because not only are they harder to find, harder to smell, but there's a very small reward. And if half the seeds are empty, that's even less of a reward. So the prediction is not only that the densities will have that kind of a pattern, basically a reverse log pattern of densities versus size, but I also make the prediction that the total seed caloric value left in the seed bank is going to be the same amongst all these different species. Because rodents are the rodents, and they're going to keep working to get that minimum amount of energy. And so that minimum amount of energy should be this. And so we're testing that now, that this number of small seeds should have that caloric value, and that number of big seeds should have that caloric value. So we have to figure out what the caloric value is first, as well as the seed densities. So that's sort of where we are. That's the prediction of how these fruit and seed get deep enough in the soil, even these gigantic ones. And as simple conclusions, there's clearly been some shifting external processes, at least in the subfamily. So you got to give me 60 million years there. Not too hard to process. We have chaparral picking up. We have shifts in climate. We have seed dispersal and seed predator shifts in these groups. And specifically in Arctostaphylos, we've got fruit dispersal that goes down rather than horizontal now. And that's reflecting a lot of changes. But the big ones are seed dormancy and the development of persistent seed banks that can survive wildfire. And then the one that permits the high diversity is probably the loss of sprouting. And that permits the genetics of these populations to drift through time. So the rodents are being manipulated by that high proportion of empty seed. That's okay. It's better than me having to put a lot of rattlesnakes out there that I might run into in the future. So here's the way I like to end things. You have a hugely diverse group, the best group in California. I actually said that in a talk once in a meeting. And the next guy said, well, mines are the best. I actually had it on a slide, so I ruled on that one. Now here's a nice manzanita plant. They're gorgeous. We've got huge diversity all throughout the state, but especially on the coast. But it turns out that as the fundamental plant for an ecosystem, they have interactions with everything. So mycorrhizal fungi, a lot of granivorous animals like rodents. And today I've just tried to pick this particular mutualism out for you because I think it's a key mutualism that has evolved the diversity of this group and permitted to adapt to a lot of different conditions in the context of wildfire. Thank you for your patience. Any questions?
Questions?
Especially from you guys who have to fill out this sheet saying that I want credit for this.
So they have a lot of similarities to Madrone, like the trees or larger species, or do they share a lot of similarities to the smaller shrubs and things like that?
The flower characters are pretty similar, but the fruits are totally different. The leaves are gigantic in Madrones compared to Arctostaphylos. A lot of different morphology changes in Arctostaphylos. They have stomata on both surfaces, whereas madrones and actually all the other genera only have them on the lower surface, and they tend to hold their leaves like this. So that means the entire water balance of Arctostaphylos has diverged away from all the other genera. So there's a lot of different physiological shifts, and that's also consistent with shifts in xylem wood anatomy. They look alike from a distance, but there are actually a huge number of differences.
I'm curious about when you were doing those caching studies, where you did those, and across the California landscape, would you expect different outcomes if you were to look at different species of manzanita with different size seeds, for example, and maybe different rodent populations in those different areas?
How much time do I have?
Just give another talk on that.
Let's be clear, my undergraduate degree was zoology, but I never took the vertebrate biology class. So my very first experiment, we went into the Gavilan Mountains, and we were up in a nice, beautiful stand. We put out all those pans and fruit and everything. It was a full moon. We had 0 visits by rodents. And so I'm sitting there in the tent watching this full moon going, I wonder if that inhibits rodent behavior. And it did. So we next went to Fort Ord under a new moon, and that's when we got all the capture. We used 2 different sizes of fruits there. We did one other experiment. We only got partial success that night. No clue why that was, because it was a new moon. So there's variability among sites. In the Sierra, there's not as diverse a small rodent population, but there are more ground squirrels and chipmunks. So they would behave a little bit differently, but they do catch.
Question also about caching behavior. I'm envisioning rodents with tunnels here and there. Are the caches in tunnels, or are they just down in dirt and reburied? Partially, I'm wondering, you mentioned that in caches, seeds at the bottom may have increased viability. I'm wondering, are these things just covered with soil, or is there airspace above them if they're in kind of burrows, or what do these caches look like?
So this will be slight speculation, but the rodents dig a hole, dump the fruit in, and then bury them in the soil. I'm sure it's not compacted, but if they survive that first year and rains come, I'm sure the soils are now densely packed again. It would take a long drive.
This whole idea about having half the seeds pass through bear's digestive system or something else to germinate, if that's the whole.
We did an experiment with seeds out of coyote dung, and there wasn't any difference.
Roman Ginka used to put them in sulfuric acid, but
Doesn't work. I've tried that too. Yeah, the seeds are sealed. Somehow smoke can get in the chemicals. There's just 2 known chemicals at the moment. Water soluble. Yeah?
In some of the areas that you're working, the diversity of the small mammals has declined. I guess that in the sandhills, for example, there's the kangaroo rat that's almost gone, right? And then there's expansion of rabbits. And I'm curious if you have any intuition, I know you haven't collected the data, but how that invasion of human-associated small mammals might affect the net germination success and all of that.
So I haven't put trail cameras up in the sandhill part, but at the Bonnie Doon site on the schist soils, we've done work. And there's a pretty dense and diverse rodent population there still, but in fact, we got pictures of pocket mice and thought we had kangaroo rats and got really excited because there was a tuft of hair at the tip of the tail. Then we saw it against the side of a known size pan. And a mammologist, well, actually it was the second mammologist who said, no, it's way too small. It's probably the pocket mice. Oh, okay, that's good. But we got at least 4 different size classes of at least 2 genera. And I went out with a guy who trapped at the Fort Ord site where we did the caching, and there were mostly Paramystic Blowy-eye, but there were also Californicus and Manipulatus and wood rats. but we've never seen pictures of true rats, ratus rats, at least. What-
I'm curious, after the Bonnie Doon fire, do we have any information about the percentage of Arctostaphylos that came back from seed versus came back from sprouting? Is it variable on the landscape due to fire intensity?
The resprouting species there is a polyploid, and they're not very good at seed germination and establishment. And I collected data from the two obligate seeders there, but not from the resprouter, because the populations are kind of small. But I could identify them now. I'll go check for you.
So from the resprouting species, there's essentially no germination from seed?
There's always germination, but it's never impressive
No.
like the obligate seeders. And that wouldn't be true of things like Paschala up in the mountains. It's a diploid, and Arctostaphylos rudis in sandy areas near Lompoc, that's also a diploid that resprouts. So the issue is that some of them are polyploids, and that's kind of messed them up a little bit. They're still functioning. So I can't help you on that one. You gonna let me out easy like this? Okay.
Manzanita different species, and I guess maybe they would be cultivars, are used in landscaping, but yet they seem to be tied to particular soil conditions. So are the ones used in landscaping, are they so genetically, flexible that they can adapt to a lot of different conditions, or, how would you describe their use in,
Okay, this is completely anecdotal.
Yes.
I have 9 taxa in my front yard. I'm on crappy coastal Franciscan shale.
Yeah, I've got 3.
Yeah.
It's like they're doing great.
They're doing great. I planted 2 Franciscana. You guys know about the Doyle Drive manzanita that was uncovered when they were going to expand Doyle Drive to the Golden Gate Bridge?
Yes.
It was extinct in the wild, they thought. Nope, there it is. It was hiding on a little traffic thing. Highways on all sides. They did move that plant, and they made hundreds of cuttings at 5 different places to make sure it survived. And I got 2 of them, put them in my front yard, and the plant said, well, thank you, even though it was May, and I wasn't watering it enough. It was like, oh, there. It's not serpentine, I'm not next to the highway. So they did fine, and I think they all do pretty well, because they're used to really nutrient-poor, bad conditions. And the only thing I've ever had to do to stimulate them was to go dig some soil or grab mushrooms from a stand.
With mycorrhizal in there. So maybe it's the case that they do well on these very impoverished soils, perhaps because other species can't survive there at all. But yet, if you put them in nicer soils, they definitely will like that.
I'll definitely be happy with it.
Okay.
Is that the same case with the myrtifolia, or does it have a problem with the fungi?
I haven't ever moved myrtifolia. It'd be nice. I'm sure it would do fine if you could get some without phytophthora. I would be afraid to move it to my front yard. Do you have some in your yard? How are you feeling about that?
I grew the Ericaceae fine as long as I sterilize the soil.
Yeah, Rod worked on myrtifolia at one point. No, you worked on
Ericaceae.
oats. No. I'm sad about myrtifolia.
I have another question. So there are a number of species in California, but you said very few, sort of, range, basin and range. But there are a lot of diverse conditions there, so why do you think that there's so much speciation? I mean, I know it's very diverse in California, but it seems like there might be more in some of the other places in the West, more species.
We're far more seasonal than the Rockies. They get a lot of summer rain that we don't. So it's a pretty stressful set of conditions. I'm sure some other genus has figured out the Rockies and has begun to diversify.
I think so thinking like Nevada, places that, the Intermountain, you know, it's sort of, yeah, I don't know. There's a lot of topography,
A lot of topography, a lot of soil variation.
but just not that many species.
Other than our Doyle Drive individual, are there other species that we should be concerned about that are such limited ranges or threatened by human actions in one way or another?
2/3 of them are CNPS listed plants. All the ones that are coastal endemics are threatened by human development because we just got rid of our coastal commission executive director. And wealthy people want to live there and then have us pay for their house to be lived for cliffs falling. Yeah, there are endemics all over the place. It's hard to come up with a problem for them. Most of them are okay because they're in out-of-the-way places.
Do you think about the pygmy manzanita and how it's faring?
It seems to be okay.
Is it high on the threatened list, or is it?
I wouldn't care if that one disappeared. Its parent is still there and it'll be selected out again. Right. And there's a coastal version that's actually better at being pygmy, but it's just a prostrate area. It's very lovely, right on the cliffs. The other thing that's nice about Arctostaphylos is I've had to go look at them all. And that means I've had to do all of these adventurous field trips. And that's the best thing that ever happened. You get up early, you drive out a couple of hours, you hike in 6 miles, you're in the middle of no place, there's no airplanes, no signs of human habitation. It's pleasantly disturbing, if you know what I mean. Mm-hmm.
Have you done any studies on how fires influence the soil fungi associated with Arctostaphylos?
Again, the killing temperatures don't go very far, and the roots, while most of the roots are pretty shallow, they have mycorrhizae when you down low, and some fungi actually produce little structures that are resistant to heat and dryness. And there's huge diversity. There is a shift. Post-fire, ascomycetes dominate, just like in conifer forests, and basidiomycetes come up as organic matter reaccumulates. But there's this whole world that we don't know very much about, and it's very high diversity. There's hundreds in a room this size of different mycorrhizal species in a stand of manzanitas. So it's open to study. You need genetics. It's the only way to identify these things. I won't bite. Thank you.
This is kind of personal, but when did you first discover manzanita? Why was it manzanita? What was the first time you saw it? I was curious.
Okay, I actually know the answer to that one. So one of my first students, third student, wanted to study life history patterns in seed banks. So I specialized in seed banks. And we wanted to look at obligate seeders versus sprouting. So I was trained in Southern California at Santa Barbara, and it's like, CNOs, high viability, easy to identify. Because at that point, to me, it was Arctostaphylos spot. You could see the red bark, it's Arctostaphylos. What species is it? I don't know. So unfortunately, my first job was in San Francisco, and there are no sprouting ceanothus within 120, 150 miles. So it's like, we'll have to work with Arctostaphylos to make this practical. So we started learning Arctostaphylos, and you get hooked. And 2 years later, that graduate student and I went to a manzanita workshop in Southern California, and we walked around, and everyone was oohing and ahhing, couldn't identify anything, and we're like, uh-oh, we can identify everything. So you learn 15 and you're in the top 6